So I'm Nate Smucker. I'm working with the Environmental Protection Agency, Office of Research and Development here in Cincinnati, Ohio. And today I was going to sort of give a high-level overview of some of the various projects and work that we're doing, where we're using diatom DNA metabarcoding for stream monitoring and indicator development, with sort of the ultimate objective being trying to figure out how it might be useful for monitoring and assessment programs. I also want to start off thanking my co-authors and colleagues too. They've been instrumental to making this work happen, especially Eric Pilgrim, who runs the DNA lab and has been responsible for generating all of this DNA metabarcoding data for thousands of river and stream samples. But also some of our partners in Office of Water with EPA and the Office of Wetlands, Oceans, and Watersheds. It's been really nice working with them and getting their perspectives and collaborating on this work to sort of steer it in various directions. Also, I should mention that for better or worse, this was reviewed by EPA and approved for publication, but these are my words and my thoughts and might not necessarily reflect official agency policy. I thought I'd lead off, though, sort of giving some context for why we're doing this type of work. I'm sure everyone's pretty familiar with water quality problems globally and across the country, especially with like nutrients, salts, and how watershed land use affects stream ecosystems and lakes. And these are important resources, right? And sometimes it's really easy to see something that's noticeably off, like harmful algal blooms or cladophora blooms covering streams. And it can have a variety of consequences for stream food webs, macroinvertebrates and fish and habitats, but also leads to a lot of socioeconomic problems too, with drinking water, aesthetics, recreation, tourism, and a variety of things. And so sort of the driving force behind a lot of the work we're doing is trying to understand what these ecological changes might be like that occur between these really nice aquatic ecosystems before they get to the point where they're experiencing some pretty major problems. Because understanding how the ecology in these streams and rivers changes gives you the context for understanding what nutrient effects might be, or how watershed conditions are affecting streams, and ultimately how you can use that information to develop management or conservation practices, as well as sort of give you a measuring stick to understand how effective different management strategies might be, or nutrient reduction efforts. And so it's really important, obviously, to be studying the biology in these systems and trying to understand how they change in response to pollution or various stressors. Otherwise, you're sort of like, well, what's 50 micrograms of phosphorus per liter mean versus 10 or 100? And so what we try to do is try to look at the biology and what's living in these streams and how they respond to these different environmental gradients. That brings me to diatoms. I think a lot of people on the call are very familiar, obviously, with diatoms, these unicellular algae, very diverse, found in pretty much all aquatic habitats. It's really nice. They're really easy to sample out in these streams. I just have a little picture of a rock up here with some of the biofilm removed. And they're also very simple to sample, quick to sample in streams and rivers. Also been known to bring joy to those who sample them as well. But they also have this long history of research behind them and their use as ecological indicators. And it goes back even farther than these papers. And these papers aren't meant to be all encompassing or anything like that, but just sort of some key ones that I thought highlight the history and the rich ecological work that's gone on with diatoms. It's a little more focused on the United States for the purpose of this talk and presenting some of the EPA research. But even going back to 1974, there was an EPA report that Rex Lau did on the tolerance of freshwater diatoms. And I highlighted a few other EPA-related papers. Jan Stevenson and others were instrumental to sort of getting this diatom work going for rivers and streams for for EPA, as well as a lot of prominent names that were key and important to the history of diatom research in America as well, that most of you are probably pretty familiar with. So here at EPA, they actually started collecting diatoms and looking into their use as indicators back in the early to mid 1990s as part of this environmental monitoring and assessment program. They repeated it out west about 7 years later. And these data led to some of those key papers that were shown on the previous page that were focused on developing diatom indicators and how they might be used in monitoring and assessment programs. And then ultimately, as part of EPA's National Rivers and Streams Assessment, they started collecting diatoms in 2008 and 2009. And then this is a 5-year cycle. This is part of EPA's bigger effort to report to Congress on the conditions of water bodies of the United States. And so if you're kind of curious about that type of work that EPA has been doing, I thought I'd throw up this screenshot of a dashboard where you can look at these various indicators in the biology and chemistry and habitat indicators that goes into those stream assessments. And I'll be talking more about the National Rivers and Streams Assessment later on. And so as diatoms have sort of grown in not popularity, but being used and sampled and collected more in the United States, around 2010 and onward, there's also been a lot of recent developments in molecular-based methods and DNA metabarcoding. And a lot of that earlier work was laying the foundation for the possible subsequent applications and how that DNA metabarcoding data could be used, right? And so looking at different genes and primer development and the methods that go into it. And then really within the past 5 years or so, especially a lot of work out in Europe, coming out of Europe, has been doing a lot of nice studies with crosswalking their historical morphological-based indices and metrics with the DNA-based approaches and comparisons for capturing the diversity, and looking at how DNA-based approaches can be used in monitoring programs. And so it was around Eric started to get into the DNA metabarcoding work in the 2013, 2014 National Rivers and Streams Assessment a little bit, but then we started to really take off with our work around 2016. And the basics were still there for us to sort of like figure it out. How do we go from taking a mixed environmental sample of paraphyton all the way through the process of extracting the DNA, PCR, the high throughput sequencing on the MiSeq, and then how to handle millions of DNA sequences from all of these samples to ultimately look at the assemblage structure of these paraphyton and the diatoms in particular, and what do we do with all of that information. So this isn't meant to be all-encompassing. I wouldn't say this was a robust literature search that I did, but I thought it might be useful to just sort of show a plot of the number of publications related to monitoring and assessment with diatoms over the years based on a Web of Science search, and then the trend in the diatom DNA-based approaches in publications over time. So more and more papers coming out every year. And we started to get into some of the work here in Cincinnati, really heavily in 2016. Just come up a little bit there. And so we're trying to get our program going, and some of these nice key conceptual papers were coming out, the future of biotic indices in the ecogenomic era, embracing environmental genomics and machine learning for routine biomonitoring, because a lot of strong work has been being done and getting published, and sort of showing the utility of these DNA-based approaches. And sort of got me to thinking, well, it's sometimes hard to get people to embrace diatoms, let alone the DNA metabarcoding, where you might not have even names for most of the stuff that's on there, but got me to thinking of like what people might look for if we want them to embrace these approaches and see that they have a sound scientific foundation supporting them. I took the slide out, but again, just sort of connecting to this historical approach of using the microscope and morphological-based identifications. I'm not vain, but this is a picture of me from grad school demonstrating really poor ergonomics at a microscope, and some of the references that were sort of classic for our lab at the time. And so we're trying to figure out, well, now instead of looking at the morphology, counting them in a microscope, getting unique gene sequence reads from environmental samples, and even if we can't put names on all of these gene sequences, in a similar fashion, we can start to build ecological information for these amplicon sequence variants, these unique gene sequences that we get. And so if we're thinking about, well, how can we make people feel comfortable, maybe less reluctant about this type of work and what needs to be done? Eric Pilgrim, I'm not going to talk much about this part, but develop the methods, the workflows, and everything for conducting the DNA metabarcoding. Most of what I'm going to talk about today deals with characterizing diatom-environment relationships and developing indicators, but I'm also going to talk about some of our work that's examined the variability over time. And we have ongoing work looking at what confounding factors are and how that might affect our interpretations and conclusions and how these are used. I'll talk a little bit to you about developing partnerships to test and refine these indicators for real-world applications that we're doing. And then some other related work I'm not going to talk about, but we are also still looking at documenting the effects of different methods, all the way from extractions to PCR to sequencing and bioinformatic pipelines, workflows, and how that might affect results and interpretations. We're also looking at actual differences in field sampling methods and storage duration effects on the DNA. But ultimately, what we want to do is sort of build a body of evidence and examine how well this is working and provide SOPs, standard operating procedures, guidance, workflows, and analytics to states and other people who are interested in using these approaches. And then lastly, ultimately, could some of these indicators ultimately be useful for EPA's national rivers and streams assessments when they're looking at the condition of these water bodies? So I wanted to start off talking a little bit about where we started. Even though this is already published work, I thought it might help sort of give the context and sort of a storyline of where we started, where we are, and where we're going. And so one of the first things that we were doing as a group was this intensive watershed study close to Cincinnati here. We were able to go out to about 25 sites and sample them 12 times during the summer one year. And this was a watershed that our collaborator, Chris Nietzsche, has been doing routine monitoring in for over a decade. So we knew these sites really well. We were targeting a nitrogen and phosphorus gradient and an agricultural land use gradient. And ultimately, we took the sequences and looked at the relative abundances of these ASVs, but for this paper, we were clustering them into OTUs at the time. And we were able to examine how these different OTUs either decrease or increase with phosphorus concentrations. We were then able to look at the relative abundances of these indicator taxa, see how they change in response to phosphorus or nitrogen and conductivity, as well as looking at some individual random forest models for all of the OTUs and sort of combining them into measures of overall assemblage change, and how these responses and where they occur along these gradients could ultimately be used to inform what might be useful nutrient management targets that could be used for protecting your best quality streams or serving as management targets to make progress toward, if that's a feasible thing in the watershed or for these streams. So that was all very high level, but I encourage you to look at the paper if you're curious about more details. This plot here is just showing all these different responses that we looked at and sort of how they line up if you rank order them across the phosphorus gradient. So something cool that came out of this study was that we've been working with the Ohio Environmental Protection Agency, which is a state monitoring agency here. And they actually used these DNA-based results and the support for these possible targets as part of their TMDL process. And for those not familiar with it, that's sort of like a water pollution cleanup plan. It's the maximum amount of a pollutant a water body can contain and still maintain water quality standards. And then there's part of this process is this loading analysis plan that identifies modeling approaches and targets. And that's where these results were used to inform that part of the work. So then they're able to then determine nutrient reduction and allocation scenarios. So it was nice to see how the diatom responses could be used in a real world application here. Well, another part of the study too was that we had all this temporal data because we sampled all these 25 sites 12 times. The assemblage structure represented changes from streams with high percent agriculture in the watershed to low percent agriculture in the watershed. But something we looked at over time was sort of, well, how do these diatoms correspond with same-day nutrient measurements, and what might affect those relationships during a typical sampling season? And one of the things we found was that in general, the nutrient relationships were mostly remaining strong over time, but high flow events led to a temporary breakdown in those relationships for a couple weeks, but then they rebounded over time. But also one of the key findings was we looked at correlations with same-day nutrient concentrations 1 to 2 weeks prior, summer mean nutrient concentrations. And in general, these diatom assemblages were doing a nice job representing the average nutrient concentrations during the span of a season, even if there was week-to-week variability in those relationships. And another way that we leveraged the sampling effort was our collaborator with Office of Water, Lester Yuan, took some nearby stream data that had high resolution flow data and then used that flow variability to better understand the diatom nutrient relationships and refine that, and saw that by building in that variability over time, strengthen the relationships with total reactive phosphorus. And then I'm not going to explain this figure too much, it's in the paper though, but he also found that typically these diatom assemblages were representing the effects of nutrients at least 1 to 2 weeks prior to the day that you go out and sample the diatoms. So a lot of nice support for this initial step here that we were making by doing this watershed study and trying to understand what was happening over time. I do want to mention too, it's not diatoms, but all of these sites that I'm talking about today, we also have bacterial data using the 16S rRNA gene. And we took some similar statistical approaches, even though none of us are necessarily microbial ecologists. But it highlights a couple things. One, you could look at different major groups of bacteria, but two, it sort of relates to how we envision keep maintaining taxonomic databases moving forward and sort of using the best taxonomy available at the time to match to these gene sequences. Could we take a similar tree-based approach? And if we have an adequate reference database and we see sort of this cluster and we have high confidence that a couple of these might be, Agnanthidium minutissimum or something, and this one doesn't have a name, maybe we put a working name on that for the time being. And the point of that being, one, continuing to try to connect to the history of diatom research and the morphology-based identifications, but also two, there's different traits that have already been established over decades of work in the literature, and can we use those as potential indicators or metrics or building indices if that was the goal of a monitoring program. So I was just using this to sort of show a visualization of that. So that's how our group got started here in Cincinnati and the type of work that we've been doing. And what we've been focusing on heavily at the moment and are working on papers is working with this first nationwide DNA metabarcoding data set as part of this National Rivers and Streams Assessment that EPA does every 5 years. And these are probabilistic sites that go through a heavily statistical process to represent the range of conditions across the United States and in different ecoregions. And then they can let you, using various statistical weights, get a measure of fair, good, poor conditions based on some of the indicators that the program uses. So at all these sites, they collect water chemistry, physical habitat, and biology. The periphyton are collected from coarse substrata, mostly on 11 transects at each site. If rocks aren't available, they'll go to woody debris, and if that's not available, they'll do sediment samples. So then we use the DNA metabarcoding lab work. I'm glossing over that. Detailed methods are in our papers, and I can follow up with anyone who's curious. But looking at the rbcL, we are now using amplicon sequence variants instead of clustering gene sequences into OTUs based on their similarities. Because when it comes off the machine, here's your sequence, here's the number of reads that had that sequence. So it makes working with new data in the future easier, and it's more straightforward. And the data summary from all of this was that there were over 15,000 ASVs. We had over 33 million reads, almost 18,500 reads per sample on average. And for all of the statistical analyses, we were using the relative abundances of these ASVs. One of the first things we did was just plot the data with a non-metric multidimensional scaling ordination. For those not familiar with it, basically sites that are closer to each other are more similar than sites that are farther apart. And then we just did some simple correlations and plotted those on here. So this first axis goes from low phosphorus to higher phosphorus and nitrogen. pH was associated with the second axis primarily, and then a bunch of cations and conductivity sort of coming off here in between the two. I'm going to focus on phosphorus, nitrogen, conductivity, and pH for today. Those are 4 variables that historically have been very important to diatom assemblages and the taxa. And so this is sort of our initial high-level analysis that we're doing at the moment. So there were some regional patterns that popped out. We saw that the northern plains, southern plains, temperate plains, and coastal plains tended to be over toward the right in that ordination, associated with higher nutrient concentrations. So here's the phosphorus concentrations, box plots for all these ecoregions and the sites within them. So those 4 regions tended to have higher phosphorus concentrations, tended to have higher nitrogen concentrations. An exception being that the upper Midwest also had higher nitrogen concentrations, despite not having as high of axis 1 scores. And then likewise, northern plains, southern and temperate plains tended to have higher conductivity streams. And if we look at axis 2, we saw some patterns here as well. The southern Appalachians, the northern Appalachians, and the coastal plains tended to have lower axis 2 scores, and those were associated with lower pH. These ecoregions also span a wider range of pH values compared to some of the others. But also you see the upper Midwest has a bit more of a range too as well. And though the Western mountains were not plotted in the ordination with this negative of axis 2 scores, they did have lower pH than some of the other ecoregions. So I was just showing that here with the yellow asterisk. And don't expect to spend much time on this because it can just blur your eyes and be confusing. The simple message I was showing here was that if we do a generalized additive model, plot all these 9 ecoregions together, some variables are more important some than others. Some have steeper relationships than others based on these NMDS ordinations. And that's all I was trying to go for here, was to just sort of visually show that variety of responses. And sort of intuitive, you would expect that. It's a very diverse setting, the whole continental United States. And so what we did do, though, to sort of get an even fuller picture is booster regression tree analysis of these NMDS axis scores as the response variable, and then conductivity, nitrogen, phosphorus, pH, and ecoregion as predictor variables. And then we also did this without ecoregion as the predictor variable. And these are pretty good models. They highlight the importance of the variables and how that relates to assemblage change in these ordinations. NMDS axis 2, pH was really important. And the ecoregion, obviously, very important. And so we've sort of done this first descriptive analysis at the whole national scale. Next, we pretty much did the similar thing on ecoregion specific portions of the data set. That's what I'll talk about here is some of these results. And the bar chart that I'm showing just is the relative importance of all of the variables, and the ones that aren't crosshatched were the ones that were the most important variable in the boosted regression tree models. These percentages are the cross-validated deviance explained. And then the colors just match. Black is for the gray conductivity, the pink is for pH, and which ecoregions. pH was the most important predictor variable. But you see conductivity was commonly the most important variable with these ecoregion specific ordinations that we did. And if we look at axis 2, we see that phosphorus, this light blue, is one of the most important variables, sometimes nitrogen, sometimes still pH. And so even though these models don't have as much cross-validated deviance explained, they're still useful to sort of understand the importance of these environmental variables, and phosphorus and nitrogen were still important, but more with axis 2 in these ordinations than with axis 1. So that's sort of an overview of the descriptive results with these 9 ecoregions. We also, what might be a little more intuitive to someone who might be interested in using these types of approaches, we also did indicator species analysis for each of the 9 ecoregional data sets. And we also used threshold we did that as part of threshold indicator taxa analysis, which is a procedure that finds the point along an environmental gradient at which you see the greatest change in the frequency and abundance of a taxon. So here's just an example of when the decreases. To the left of this change point, you see it more commonly observed, higher abundances. To the right of it, less. Here's one where to the left of the change point, you don't see it very often. You see it in lower abundances. To the right of it, you see it more often, higher abundances. And this approach uses 1,000 or 500 bootstraps. And so you get a bootstrap distribution of all of these change points that a taxon has. Then all of these taxa change points can be plotted together. You can see the blue are the ones that are decreasing with phosphorus in this example. The red are the ones that are increasing. You see the bootstrapped distributions of these change points. And something that seems to keep reemerging in our data sets, the watershed scale, these regions, is that we often see clearer, more definitive, quick changes in low nutrient taxa. And you see them gradually replaced with more and more high nutrient taxa. And these taxa are sort of like a gradual increase. It's not like a boom, we're suddenly relieved from nutrient limitation, now's our chance type of thing. They're just sort of gradually increasing as phosphorus in this example increases. So all of these taxa change points can be combined into a number that represents overall assemblage change and where along an environmental gradient you see the greatest change occurring. And that's all this is showing is the bootstrap distribution of portions of the phosphorus gradient for the northern Appalachian streams, and where you're seeing the greatest change in assemblage occurring for the low nutrient taxa and the high nutrient taxa. And so what we could use these results for too is to just plot them all together. Here the box plots are the distribution of phosphorus concentrations in each of these ecoregions. They're just color-coded. Orange is high, gray is medium, blue are the lower nutrient ecoregions that are typically the mountainous ones, Western Mountains, Southern and Northern Appalachians. I thought something interesting, although I got to sort of think more about it, is that these low phosphorus change points across these ecoregions, despite ecoregions that have high nutrients or more high nutrient streams, ones that have lower nutrient streams, they're in a relatively narrow window here where you see a noticeable change in the low phosphorus taxa. Whereas the high phosphorus taxa, again, these are on log scales too, so these are pretty wide confidence or bootstrap distributions of change points. Those I think sort of pick up on just sort of the distribution of those higher nutrient conditions among the nutrient or among the streams in these different ecoregions. And so I highlighted in yellow the range between the lowest and highest change points in these ecoregions. Something else that I thought would be interesting, or maybe hopefully you find it interesting, was to take what we found with this one analysis. Again, this is just one part of the puzzle. You do some other analyses and they come together to sort of tell a complementary story to sort of get a clearer picture of what's happening. But here are the same change points from the previous plot shown in black now instead. And then all of these vertical bars that are red with the black line across them, or gray with the black line across them, are literature-based numbers that represent ranges of phosphorus concentrations that diatom responses were observed. Almost all of these are morphology-based, microscopy-based studies. One was not. This is our Little Miami River watershed study in Ohio that I talked about earlier. And that's part of the temperate plains ecosystem. And these are the low phosphorus change point diatoms. And the main purpose for this plot was simply to just sort of show that we're not coming up with anything that's too wild or way out there compared to what has been reported in the literature. And we also had another nice diatom study that also used threshold indicator taxa analysis for Connecticut streams years ago, and that was morphology based, and it had some actually pretty similar change point distributions to the Northern Appalachian data set as a whole. And there's a couple of studies that were done in the mid-Atlantic, Hausman, Stevenson, some out west, the USGS has been involved with, Ian Waite, Mark Munn. So again, I'm just sort of pulling together different pieces of the picture here and just sort of seeing how what we're doing is comparing to what was already has been found in the literature. And there might be studies I've missed, but it was sort of promising to see this. So something else as a way that we're approaching the data and how it could be used is some of this might seem a little like, okay, how do I use this if I'm interested in a monitoring assessment program? Like, am I supposed to look at every ordination individually? Am I supposed to run a threshold indicator taxa analysis with each unique data set? Or can we use this information to start to build traits and ecological information for these amplicon sequence variants? And so that was the next step. We looked at all these ecoregion specific indicators and then examined whether or not the relative abundances of these indicators can be used and how they are associated with the environmental variable of interest. So this is an example, Northern Appalachians decrease in low phosphorus relative abundances as phosphorus increases, increase in high phosphorus ones, although that's a pretty noisy looking relationship, but it also sort of is nice because it's not like it's a one-to-one trade-off between, well, if the low nutrient ones decrease, high nutrient ones just automatically increase or something. So it's not necessarily just an inverse relationship, and it might be additional ecologically informative information to be gained. Similar to the previous plot, again, nothing to dive too deep into here other than these are showing all 9 ecoregions, relative abundances of high phosphorus and low phosphorus diatoms as the example, and how those relationships are different among the regions. So some of these regions had really high phosphorus concentrations, some had lower ones. But in general, we do see, and I wasn't going to show a whole table of Spearman correlations, but they did consistently have correlations with phosphorus and the conductivity indicators with conductivity, but some of them not as strong as others, right? So this is the coastal plains here, this dark purple line, not much of a slope, pretty low relative abundance of low P diatoms, but it also is an ecoregion that tended to have higher nutrient concentrations. And if you look at its change with the high phosphorus diatom relative abundances, it was a pretty steep, strong relationship. Some of these, I should mention, look hump-shaped, but that's a data distribution problem. So this is the Western Mountains. There's not actually much data out this way. You can see this really wide confidence interval for that one. And so that's just something to keep in mind if you're thinking about this later when you look back at the recording. And so based on correlations, just sort of this initial sort of crude look at it all, it shows that there could be potential here with using these as metrics and indicators that could be useful from a monitoring perspective. Something else, I talked a little bit about temporal variability with the Little Miami River watershed study that we first started with. And as part of the National Rivers and Streams Assessment, they do go back to a subset of sites twice. And so for 183 sites, they went back a second time. And what I'm showing here is the Bray-Curtis dissimilarity between all pairs of sites and the Bray-Curtis dissimilarity within sites for those 2 visits. And you can see that pairwise, sites aren't very similar to each other in general for the most part. Within site, you'd probably still say, oh, these are still pretty different between the 2 visits if this is 50% dissimilarity, and sites over here are more similar between the 2 visits, sites over here less similar between the 2 visits. And if you want to just sort of plot them together to sort of see how that looks, you're like, okay, so there's within site is more similar than among site, which is great, but then you might be sort of hesitant and wondering like, well, if you think of like microscope counting slides and 60% similarity being a potential QA/QC target, not a lot of these hit 60% similarity. And a part of that might also just be an artifact of how we were putting the data in and which taxa were included. We weren't diving deeper yet into the diversity or the evenness of these samples. But something interesting, brace yourselves, is that if you look at the metrics, the relative abundances of low phosphorus, high phosphorus, conductivity pH, nitrogen, if you look at those and see how those compare between the 2 visits, you actually see that in a lot of these ecoregions, which are the columns, a lot of them were actually pretty similar between the 2 visits. There's some that are pretty different. I mean, these are noisy, transient systems, right? And things happen between a few weeks sampling that could lead to big differences. There could also be just innate within site differences in where the samples were collected potentially. But I thought it was interesting to see this plot, even though it's a lot to digest now. Again, feel free to look back at the recording and pause it to look longer if you want to think more about it. But it was sort of interesting and good to see that these relative abundances of the indicator taxa seem to be consistent for the most part. We obviously need to put a little more quantitative number on this because sometimes they're pretty different, other times they're very similar. But I thought it was at least like sort of a useful first approach to sort of seeing how similar and different they might be between visits. So that's where we are. We're working a lot with the NERSA data set. Lester Yuan's leading up a really nice effort looking at beta diversity and species richness and diversity patterns in the national data set as a complementary part of this work. But where we're going is we've sort of got data at different scales already. Experimental mesocosms there are grayed out because we have the data, we just haven't analyzed it yet. We had our watershed study, we're working on the national study, but we sort of have this gap between watersheds and the national scale, and that's the states and getting a fuller regional representation. And so over the past, really this year, we've simply just been trying to reach out to potential partners and see if they're interested in teaming up. Some states like Indiana, Wyoming, Connecticut, Maine already have state-associated diatom monitoring practices going. And so we're like, hey, if you're out there, you're grabbing paraffin, you just like split it in half, freeze it, and send it to us to work on and see what we find. And some of the main reasons for this is that it fills that gap between watersheds and the regional national scales, but it also is a chance to look at these different state monitoring assessment programs and see, okay, what are the potential real world applications and use of this work? And if we get new data sets, it allows us to test and refine the indicators and the work that we're doing, seeing how they work, or maybe they don't work all that great. And you can dive into like why, or what can we do to make it better, and things like that. So it's been exciting. Sylvia Lee has been really important to the Midwest and leading a project there that is looking at diatom indices, and we're going to be doing some DNA metabarcoding work for that for Wisconsin, Michigan, Indiana, Illinois, and Ohio. Got some great partners in California with the Southern California Coastal Water Research Program that are really like driving their own research and development with stream monitoring assessment approaches with DNA-based data. But we're looking forward to working with these states. And we've also been like, hey, maybe there's some people out in academia that have some study systems. And if you're interested, we would also be interested in hearing about that and seeing if there's a potential for leveraging existing efforts to try and get this to a point where it's usable and defensible and reliable. And so as part of that, and sort of wrapping things up, we have these different studies, right? And it's a lot of data to manage. We got 4 years of nutrient dosing experiments in these mesocosms. We went back out to the watershed to do a storage duration study. We're looking into partners to take raw samples all the way through the process from extraction to sequencing, bioinformatics to see if everybody's arriving at the same general picture. I know there's already been a big scale effort that's been done, headed up by some people from Europe. As a part of that, so something similar. And then we also have an urban gradient study that we're looking into, that we're working on from Southern New England. And then we have all these state-specific studies, and a lot of these are states that have rotating water basin programs as part of their monitoring program. And so every year they hit a few basins, and then new ones the subsequent year, and they just cycle through them. And so how do you handle all of that? And I know that I feel like historically, people have had hesitancy over some statistics they view as being a black box, but what we're trying to do is build a black box that can take a new data set and funnel it through an R workflow and R scripts and merge it with existing data sets and continue to grow those data sets, like this database where we can keep track of all the ASVs, the metadata, the studies that they were first observed in and where they came from, start to build the traits databases there and update as well as any taxonomy that we're able to assign. We want this to be able to just basically like a click and go, put it through the analysis if you're interested in ordinations, generate metric scores for your sites or your study, get box plots or scatter plots, things, and other ways to visualize that data in an efficient manner. Otherwise, it would be sort of like, okay, we sent this off to a commercial lab and we got our table back of gene sequences and the number of reads in the sample, now what type of thing. And so we're hoping this will sort of ease any reluctance or increase the interest that potential monitoring assessment programs might be interested in using this type of work, but we still have a ways to go on that. And then just putting it up for this last slide is just sort of the overview of the process we're going through to look for these responsive indicators, build partnerships with states and other people, continue to test and improve these DNA-based approaches, including a fuller analysis of these indicator taxa to better understand any cross-correlations or effects of confounding variables. And then also linking these to species names as best as possible and relating them to traits, and also the work we're doing to just sort of characterize the diversity and distributions based on this. Although I will say this is obviously just like based only on the rbcL gene that we're working with. Just to keep in mind there when we're thinking about diversity and distributions. And with that, just want to acknowledge especially all the field crews and the state agency people who are out there making all of this data possible, as well as Sarah and Barry, who do a lot of work in the lab for the DNA aspects. And Tracy Schiffling has been a big help with the the R code. And then nobody asked, but I like this diatom. It's dose of nostalgia for the first diatoms, and I really like focused on a little small study 15 years ago. I'm just like, oh my gosh, that's 15 years ago. But Thalassius lacustris that Mark Edlin so generously teamed up and let me nag him over for year or 9 years, but a long time to focus on that little paper. So with that, I'd be happy to answer any questions or hear your thoughts or just have an open discussion about anything.
Hey, Nate, that was really, really great. And I'm familiar with some of your initial work on this, but it's really great to hear what you guys have been doing in the last few years. I feel invigorated to go and tidy up one of my dissertation chapters and get that out there to the world. So thanks. I wanted to ask you, and again, if anybody has any questions, please feel free to drop them in the chat or raise your hand and ask Nate directly. But what kind of have you ran into any roadblocks and pursuing using ASVs and DNA as biomonitoring tools? And if you have, what has helped you overcome those?
No real roadblocks at the moment. And I should also say, I mean, we're obviously still, while we're doing a lot of work, it's still early on in the process. But it seems more straightforward and reproducible and consistent moving forward to work with them. And I skipped over that part of it too, right? Like, I mean, we have Eric Pilgrim does a lot of the front end work to like clean up what comes off the sequencing runs and getting rid of any sequencing artifacts or chimeras and things like that. Yeah, so it's sort of, it also seems to be the general direction that the literature is moving in, the amplicon sequence variants, as opposed to clustering into OTUs.
Great, great, thanks. Hi, Ben, first time meeting you. You have a question for Nate?
Yeah, thanks for having me. Nate, we obviously talked at the National Monitoring Conference, but I was just curious, you mentioned some R scripts and the data analyses that you all have been working on. Is that anything is that in development, or are some of those scripts currently on like GitHub or something that could be reviewed and tested?
Well, that's a good idea to think about moving into the future. They're all in development in-house right now. Yeah.
Cool. Good to know. Thanks.
Ian, I see you turned on your camera. Does that mean you have a question?
Yeah,
or is that an accident? Okay.
Hi, thanks for a great talk. I was going to ask if you could just comment. Sorry, I have this going on two screens. I was wondering if you could comment on some of your, well, maybe it's too nitty-gritty, just thinking about your results in the context of your relative abundance analysis, if you've looked at frequency of occurrence, presence, absence, just thinking about the compositional elements of this data and wondering if you've seen different things or thought about that in any particular way.
No, that's a really good question. And so I have been working solely on the relative abundance aspect of it, but our colleague Lester has been exclusively taking presence absence approach. And it's been interesting. I mean, with the watershed scale and the national scale, that they seem to be arriving at similar pictures just in a complementary type way, especially with the aspect of that he's been looking at, like some work looking at niche breadth and distributions and beta diversity patterns based on the presence absence. And, you know, it's something that's sort of on that long list of things to get to at some point where we could see, okay, if we just put things through side by side, presence absence, relative abundance for the same exact, well, where you can, a similar type of analysis, what do you find? Yeah, that's been the extent of it so far, though.
So, Nate, I was also curious, you mentioned just somebody freezing some paraphyton scrapes and sending them to you as far as, you know, partnering with folks and collecting. you know, we've experimented with using Zymo and ethanol and different sorts of DNA preservatives. Are you literally just freezing some periphyton samples and working with those? Do you find you get good results?
We are, and we do. And I think there's been hesitancy, depending on like sort of the scale of the work as well, with what it's being preserved with for shipping and how well it preserves the DNA and handling it on the back end. And so it has been something that Eric's lab has just sort of been set up to handle since he's taking on 1,000 samples a year as part of the National Rivers and Streams Assessment, that it's sort of what's feasible, I think, and cost effective maybe. And we do have that small study where we are going to look at freezing duration effects on results, and we'll be able to look at presence absence of what came off or came out of it, as well as the relative abundances of those indicators. Does it affect relationships with environmental variables? But we haven't started to work on that yet as far as the data analysis goes. But yeah, so it has been, I think, effective and useful, but obviously it's always good to like double check everything, right? And see if you're disproportionately losing some taxa consistently or something because of a certain storage method. Yeah, no, good question.
Thanks. Richard, do you have a question?
Just more to follow up on Nate's point there, that given, at least for the national surveys, given our logistic issues, at this point in time, we've decided to go with freezing on dry ice and shipping that way. But those have been discussions we've had, you know, whether or not we should be filtering it in the field or waiting for Eric to filter it and things like that. So I think we, as Nate said, we probably want to do more on that, but we seem to be still getting pretty good results with the approach we're taking. So, but yeah.
Thanks. Yeah, I'll take that and put it in my notebook. I really appreciate that sort of feedback. I know some of the work in Europe has shown that different diatoms, different size diatoms, contain different copy numbers. Have you guys done any efforts to come up with or use correction factors when working with these relative abundance numbers?
Yeah, so very good point. And yeah, you've seen those studies. And it is an interesting thing. We have not been doing any corrective factors because one, also, I think they've been finding a relationship with biovolume as well. And that's sort of kind of interesting because then if your abundance of gene reads are sort of connected to that biovolume in a way, even if you don't know exactly, it sort of makes a connection to the biomass in the system. That's also potentially why there's an argument for presence absence versus the relative abundances. Yeah, I am not well versed in it, I would say, but I think a problem is just sort of, there's a lot of species of diatoms, and how are you getting a good measure of their biovolumes that you're going to get out of the sequencing run to make that connection? And that's sort of something else that I think is sort of a concern or something to think about with like sort of those morphology to DNA comparisons to that are made directly. But yeah, no, that's a good point about the number of gene copies, the relationship with biovolume. And it is something that's been interesting to see with Lester's work that's been heavy with the presence absence approach, telling a complementary part of these stories when it comes to the environmental variables that are important in nutrient relationships. So it's kind of promising. I mean, I get it. We don't want to put the cart before the horse, right? Or whatever the saying is. But we are also just sort of like, well, we have the data, let's work it through and see what we find. And it's making sense, I think, from an ecological perspective. And we're continuing to find that it keeps working.
Great. Nick Schulte has a question he wrote in the chat. Do you see taxonomy-free based analysis as the way forward for NERSA and state surveys, or will you look into taxonomic assignments as well?
I think it's always good to continue to do the best you can with the taxonomic assignments, just because that gives you more information to work with and more understanding of what you're doing. At the same time, and again, so I'm not recommending anything, but I can say that this is what we've been finding with our studies, is that the taxonomy-free approach is okay to proceed because you're continuing to sort of build that understanding in the ecology for these ASVs, regardless of what species name you put on them, or whether or not you find a match in a taxonomic reference database, right? Because I know that's a problem that I think plagues a lot of people's work is, I think we find less than 40% names on things, and that's even less if you want to go with like something that's a more confident name to put on it. And so it's sort of like, well, work with the ASVs, taxonomy free based, which is what we're doing, but then it's sort of then somewhat disconnected from all of that rich history of diatom work that has worked towards understanding their ecology based on what's been a species in the past and connecting it to the DNA data. But I don't think they have to be exclusive. I think go forward as best you can with the taxonomy free based approach and continue to try to grow the taxonomic information as much as you can as you go along.
Awesome, Nate. Well, thank you very much. I see some applause there popping up. And out of respect for everybody's time, I think we'll draw to a close for now. If anybody has any more questions for Nate, I'm sure they could reach out to him or reach out to me to get in touch with him. Really appreciate you chatting with us today. On November 7th, we're going to have you and Revi talking about something that makes the Great Lakes so great. Guessing that's going to be some more diatoms. And thanks again, Nate. Really appreciate you having us here today.
Yeah, I appreciate
being here with us today. Thank you.
being here. Thanks. Happy to be here. Take care.
Thank you.
Thank you.